Good morning, everyone. I am your guide for today, together with my colleagues, to introduce to you the concept of cells. First off, we are going to tackle about the mastermind or the theory that brought us to this interesting field of study. So, what is the cell theory? The cell theory is a unifying principle of biology that states that all biological organisms are composed of cells. Cells are a unit of life. All life comes from pre-existing life. The cell theory is so established today that it forms one of the unifying principles of biology. It is developed by the following scientists named Matthias Schleiden, Theodor Schwann, Rudolf Virchow. In 1665, Robert Hooke published Micrographia, a book filled with drawings and descriptions of the organisms he viewed under the recently invented microscope. The invention of the microscope led to the discovery of the cell by Hooke. While looking at a cork, Hooke observed box-shaped structures which he called cells, as they reminded him of the cells or rooms where monks lived in. This discovery led to the development of the classical cell theory. In 1675, using single-lens microscope of his own design and make, Antoine van Leeuwenhoek was the first to observe and to experiment with microbes, which he originally referred to as Dierkens, or the German term for animalcules. He was the first to relatively determine their size. Most of the animalcules are now referred to as unicellular organisms, although he observed multicellular organisms in pond water. He was also the first to document microscopic observations of muscle fibers, bacteria, spermatozoa, red blood cells, crystals in Gotoffy, and among the first to see blood flow in capillaries. Although van Leeuwenhoek did not write any books, he described his discoveries in letters to the Royal Society, which published many of his letters and to persons in several European countries. In 1838, Matthias Schleiden, a German botanist, concluded that all plant tissues are composed of cells and that an embryonic plant arose from a single cell. He declared that a cell is the basic building block of all plant matter. This statement of Schleiden was the first generalization concerning cells. Schleiden investigated plants microscopically and conceived that plants are composed of recognizable units of cells. He thought plant growth came about through the production of new cells, which he speculated came from the nuclei of old cells. Although later discoveries proved him wrong about the role of the nucleus in mitosis, his conception of the cell as the common structural unit of plants shifted study from two processes going on in a living cell. In 1839, Theodor Schwann, a German biologist, reached the same conclusion as Schleiden about animal tissue being composed of cells, ending speculations that plants and animals were different in structure. Schwann described cellular structures in animal cartilage. He pulled existing observations together into a theory that stated cells are organisms and all organisms consist of one or more cells. The cell is the basic unit of structure for all organisms, and plant and animals consist of combination of these structures. In other words, the cell is the basic unit of life. This statement was the second generalization concerning cells and is the most important in the development of biology. It became known as the cell theory. And lastly, Rudolf Karl Virchow, or Virchow, in 1835, the German physiologist, physician, pathologist, and anthropologist Rudolf Virchow was able to add a third part to the cell theory. He stated that all cells develop only from existing cells. Virchow was the first to demonstrate that the cell theory applies to diseased tissue as well as to healthy tissue. Diseased cells derive from the healthy cells of normal tissue. He did not, however, accept Louis Pasteur's germ theory of disease. Since the formation of classical cell theory, Technology has improved, allowing for more detailed observations that have led to discoveries about cells. These findings led to the formation of the modern cell theory, which has three main additions. First, that DNA is passed between cells during cell division. Second, that the cells of all organisms within a similar species are mostly the same, both structurally and chemically. And finally, that energy flow occurs within cells. To continue our tour, let me show you the three types of cells.
There are three types of cells, namely plant cell, animal cell, and bacterial cell. Plant cells are basic unit of life in organisms of the kingdom of the Plantae. They are eukaryotic cells which have a true nucleus along with specialized structures called organelles that carry out different functions. Animal cells are also eukaryotic cells. Animal cells are the basic unit of life in organisms of the kingdom of Animalia. Bacteria are single-cell organisms that are neither plants nor animals. They usually measure a few micrometers in length and exist together in communities of millions. A gram of soil typically contains about 40 million bacterial cells. A milliliter of fresh water usually holds about 1 million bacterial cells.
There are three types of plant cell. Let me introduce to you to each of one. First is parenchyma cell. Parenchyma is a type of simple permanent tissue that makes a major part of ground tissues in plants, where other tissues like vascular tissues are embedded. They are non-vascular and composed of simple, living, and undifferentiated cells, which are modified to perform various functions like regeneration and wound healing. Other key functions of these cells include photosynthesis, storage, secretion, and transport. Second is collenchyma cells. Collenchyma cells are elongated cells with irregularly thick cell walls that provides support and structure. Their thick cells walls are composed of the compounds cellulose and pectin. These cells are often found under the epidermis or the outer layer of cell in young stems and in leaf veins. It provides support, structure, mechanical strength, and flexibility to the petiole, leaf veins, and stem of young plants, allowing for easy bending without breakage. Lastly is sclerenchyma cell. Sclerenchyma cells have thick, lignified secondary walls, lack cell contents and maturity, and occur throughout all plant tissues. These features make sclerenchyma tissues hard, rigid, and somewhat brittle. Sclerenchyma cells can occur as aggregates within ground tissue.
Hello everyone and welcome to the endomembrane system. What is the endomembrane system? Well, it is a functionally interrelated membrane system including the endoplasmic reticulum or the ER, the nuclear envelope, the Golgi apparatus, vacuoles or lysosomes, endosomes, the plasma membrane, and vesicles. The endomembrane system comprises a complex network of membrane trafficking, which is essential to the transport and exchange of materials such as proteins and lipids. There is no doubt that membrane trafficking fulfills a fundamental function, supporting cell proliferation, maintenance of cellular homeostasis, and specific demands for higher order function in multicellular organisms. Organelles collaborate to carry out certain jobs and fulfill specific responsibilities in order for cells to operate and thrive. Each organelle performs an essential function in the cell survival. This is called the division of labor. On a more granular level, each type of cell in a plant is divided into a number of specialized compartments, or organelles, each with its own membrane. The nucleus, which contains the genetic information required for cell growth and reproduction. Mitochondria, which are responsible for the energy transactions required for cell and organism survival. Lysosomes, which digest unwanted materials within the cell, and the endoplasmic reticulum and Golgi apparatus, which process, sort, and direct molecules such as proteins to their proper locations in cells and tissues.
And that will be all for our cell tour. I hope you enjoyed and learned a lot. See you on the next one.